Thank you very much. It's a pleasure to be in Sofia. I left Nigeria yesterday. Nigeria is 32 degrees Celsius. I get to Milan. Milan is 10 degrees Celsius. And I got to my hotel like 2 a.m. this morning at -4. So Hesham told me the next place to go is Russia in Siberia at -30. I said, no, the next place is to go back home. So it's a pleasure to be here. Thank you, Maya. That's the advantage of mobility in life. We met 2010, 2011 in Germany when we were both Humboldtians. And when it was over, everybody came back to Bulgaria, went back to Nigeria. And then the Humboldt colleague in Bulgaria was 2024. That's 13 years later. And she remembered me and said I should be invited. So nothing is lost with mobility. Sometimes we get the advantage of network immediately. Sometimes it's 10 years time. So we just keep doing what we need to do. We may never know the profits. So today I will give a short talk on active surveillance of endemic and emerging viruses. I'm from the University of Ibadan in West Africa in Nigeria. It's the oldest university in Nigeria. So 1948, I'm sure that's very young to compare to some other universities in Europe. We have one Nobel Prize winner from the university in literature, Wole Soyinka. In the Nigerian Academy of Science, Nigerian Academy of Science appoints 10 people every year. 10 top scientists are elected into the academy. It's never more than 10. So I was elected in 2024 into the academy, and we were 9 of us because the 10th position was tied, 2 people, and they never award more than 10. So they dropped the 2 of them and took 9 of us. So the University of Ibadan has the highest number of people in the Academy of Science, and we have a long history of biomedical research collaboration with Wellcome Trust, with the NIH. And so it's a lot of research-based university. Yeah, of recent, because of economic downturn, it started to affect the quality of research. Top scientists, top researchers are leaving the country and going to the US and go. But we are hopeful that we'll be able to redeem that over time. So in 2021, the Humboldt, as the pandemic was coming to an end, it was obvious that Africa didn't respond well during the pandemic. Surveillance was low, research was low, so their contribution to even diagnosis was also very low. And the pandemic told us that if I solve my problem and my neighbor still has a problem, it's just a question of time. I'm going to get the problem again. And I think the Humboldt Foundation decided to sponsor six hubs in Africa, 5-year funding, 750,000 euros. So we're told to write our proposals, and they gave some criteria. You must have been a Humboldtian. You must have gone back to your country. You must be a full professor. They had some criteria, and I was able to tick all of them and then wrote the proposal. And we were told that 21 people were qualified, wrote a nice proposal, and at the end they picked 6, and we were among the 6. So you must have a German collaborator. So we collaborate with the German Center of Excellence for Infectious Diseases, Friedrich Loeffler Institute in Greifswald. And then I have a co-Nigerian investigator, Christian Happy, who is a very famous virologist. Okay, so going to the lecture proper, what is becoming obvious in the world is that viruses are not going away. It's obvious because there's climate change. It's obvious because human activities is busy bringing down forests, encroaching into wildlife habitats. It's obvious because human beings are having risky behaviors. We're getting too close to animals. And so with that, you are having a mixture of genes, humans to man. There's a lot of mutation going on. We are also performing some risky research, gain of function. So there's no doubt that we are having a future ahead of us that we need to prepare for today. I'll get to it. One obvious one we've seen is the bird flu moving from birds to cattle to humans and becoming infected. And as a veterinarian, I'll say, well, cattle, because it was obvious from they stopped milking, they became weak. The milking industry is a big one, so they investigated. How are we now sure that the bird flu is not with dogs, is not with pigs, is not with other animals? So active surveillance, checking the human and animal population for emerging and re-emerging diseases, very important if we are going to develop vaccines in preparation for future pandemics, if we are going to predict future pandemics that can attack us, and to know the risk that we are finding ourselves. So climate change, human habitat mutation, and the ecosystem. When I was in high school, I used to say the food chain, you have humans up, then you have animals, then you have low animals, then you have bacteria. But now we all live together. So that's gradation doesn't seem to exist anymore. So bringing new challenges to disease risk and disease spread. So when we started this program in 2021, the first thing we started with, then I was still the director of the veterinary hospital, was to look at the dogs that were coming to the clinic, if they were performing of any epidemiological importance in COVID. So we had ethical approval. We took consent from dog owners, and we're able to look at close to 200 dogs that came to the clinic normally for deworming. They came to deworm their dogs. They came to vaccinate their dogs, and we're able to take serum samples. And 12% of the dogs that we examined with the rapid kit test were positive for COVID. Now, none of them came, I think, of the 23, was only, when we went back to the records, only three or four of them had fever, but 23 of them were positive. The clients came for routine deworming. They came for routine vaccines. And so with the rapid kit test, we took it to the PCR, and we see the confirmation that these animals actually had COVID. Now, the viral load was low, but by the time we went back in 2023 to look at some of those dogs that were positive, we were able to get about 6 out of the 23. And when we looked at the 2 were still positive, whether it was a new infection or they remained with the infection that they had, we were not able to discern, but 2 of those animals still proved positive. 2024 went back to those 6 animals, 3 remained positive for COVID. Like I said, we can't discern whether it was they were reinfected or they remained infected. Now, the sad part was that, particularly the households, there was one dog that throughout had been positive, but the owners of the dog, the family, father, mother, 4 children, refused to take the COVID test. They said they were able to cooperate with us for their dog, but they are not ready to cooperate with us for themselves. And so the role the dog was playing in maybe transmitting COVID. But one thing that was obvious from my experiment was that dogs could be reservoirs for the COVID infection. They had low titer, but the low titer can be maintained over a long period of time. So when the dog becomes immunocompromised, okay, maybe it has babesiosis, it has bone marrow depression, what will be the rate of growth of such a virus? Or if such dogs were in contact with immunocompromised people, what role could they play as reservoirs? I think in the same vein, we have 19 monkeys non-human primates in the zoo. We have the biggest zoo in the country, and we went through a special ethical clear approval to bleed the zoos during that period. And of the 19, 3 of our animals were positive for COVID. So now COVID-19 seems to have gone, but we should be concerned about animals that could still maintain reservoir status of the disease. I think we have this. Yes, we have this submitted to BMC Veterinary Research, the dog walk, and we already received the first reviewers' comment, which we have corrected and sent back. So we are very hopeful within a month that we'll have this work published. So that's about COVID. Then let me go to an endemic disease, African horse sickness. It's every year, November, December into January, we always have an outbreak of African horse sickness in Nigeria. Some of them are particularly during the polo season. So when the horses congregate, and we decided to, about 1984, we had a comprehensive surveillance on seroprevalence of African horse sickness in the country. Some work about 10 years ago, but 50-something animals, so we didn't have enough number of observations. So we decided we looked at almost 600 animals, horses across the country, across 6 different regions, and 90% seroprevalence of African horse sickness. So it's an endemic disease, which we are concerned about, mostly with older animals, okay, more in females than in males, but for the sake of unusual hosts and the epidemiology of the disease, we decided to investigate the seroprevalence of African horse sickness, not just with horses, but with wild rodents, wild animals in general. African giant rat, the bats, cattle egrets, crows, hedgehog, pigeons, and squirrels. But I mean, good to report that there was a 0% prevalence. So at least African horse sickness is still traditionally a problem with horses in Nigeria, although in South Africa, it's been reported that dogs also harbor the virus for African horse sickness. So is this one of the most comprehensive work from across the country and with high number of observation. This work is submitted this to Veterinary Medicine International just about a month ago, early this year. So we are hopeful that we can get this information out about this endemic disease. So I'll go to Crimean Congo, work we've also been doing in the lab. This is a virus we've been working on in Nigeria for decades. And first and foremost was also to look at Crimean Congo and Nigeria in unusual hosts: pigeons, crows, cattle egrets, bats, hedgehog, pangolins, primates, and we also saw virtually a 0 prevalence in Crimean Congo in unusual hosts, but high prevalence in horses. We also just got a recent data for small ruminant sheep and goats, so have a high seroprevalence in Crimean Congo in sheep and goats. So that's the work we've done so far. We're now going to run the PCR with the horses and with the sheep and goat. But our most comprehensive work for Crimean Congo has been done in Benin Republic with our neighbors that we are partnering with in the University of Abomey-Calavi. And they were able to go to about 15 regions of Benin Republic and bleed animals, both domestic and unusual hosts. And so far, the results in wild animals shows a very low prevalence. Similar, we've got virtually 0 prevalence in Nigeria. So less than 2%, about 1% prevalence in wild animals, just one case in squirrel, one case in. So we are on this of the you have a with And those are one of the things that made us write this proposal. Is traditionally in the last 20 years, when they mention diseases, Crimean Congo, influenza, they look for seroprevalence in different countries. When you get to the WHO data, when you get to Nigeria, data not available. Data not available. That has been the tradition for many years, and that's a narrative that we are willing to change. Or sometimes when we were taught in school, they'll tell us that disease does not exist in Nigeria. And the question is, did you look for it? If you don't do surveillance for something, the fact that you do not see obvious signs, you can't conclude that it does not exist. I mean, we still say mad cow disease does not exist in Nigeria. You can't say that. You have to make the effort to do an active surveillance before you can rule out that disease does not exist. So this was the first report of Crimean Congo in Benin Republic, which is quite very important. And the detection that Crimean Congo exists both in domestic and wild animals, even though the prevalence is low, is significant as a public health threat. And this underscores that we need to build capacity. And by the time we do the sequence of the virus, we'll know whether we are having a unique type or probably where is the molecular evolution from. Is it typing with the one in Europe, typing with the one in South America? Try to have an idea of where such diseases have come from. We've also submitted this to viruses, journal viruses, seroprevalence and risk factors of Crimean Congo hemorrhagic fever exposure in wild and domestic animals in Benin in West Africa. Okay, I think I have two more diseases to go to. West Nile. So West Nile is a disease that's quite important. It's one of the most disease with highest spread in Europe. And so we decided to look at West Nile in Nigeria since 2022. And so we went through bats, non-human primates, crows, hedgehog, squirrel, African giant rat, pigeon, cattle, egret, pangolin, and unlike what you have in Crimean Congo and African house sickness, there was no species that did not have seroprevalence for West Nile. Or particularly the primates was 74% of the primates that we investigated was positive out of the 19, like I told you, I have 19 monkeys in the zoo. So this really caught our attention, and we decided we are now spending far more time now on West Nile in the lab. I have 2 PhD students working on West Nile because it's obvious that this really is a threat. This is in the system. This cuts across species. This definitely is a virus of pandemic potential and that can spread around and affect the whole system. So this is the work in the monkey. I said to us in the non-human primates. This is the ELISA, and then we run the PCR. So when we did the PCR, we had almost 50%. And then 1 year later, we went to bleed the animals again. 1 year after, this was 2023, is 2024. By 2024, virtually all were now positive. So but these monkeys have no clinical signs. Temperature is normal, they don't have fever, and then the few that have died, that we posted them, because in the veterinary hospital, one had a stroke, the other had pneumonia. So you couldn't really pinpoint the death to West Nile. And the PCR actually also shows very low titer. So it's there, it's maintained. The titer is low, but it's not eliminated. 1 year after shows that. So we ran the sequence for the West Nile, and it's 99% similarity with lineage 1, which is the lineage we have in Europe. So West Nile it's a problem, and because West Nile has it's flavivirus-like families, they have genetic similarities. The West Nile virus itself, the St. Louis encephalic virus, Zika virus, Usutu, dengue. So you have that. So we are expecting, we expected it in December, but I think we're having problems with export permit. So we are getting the primers for all these identical viruses. We're getting the primers from Germany, from the Friedrich Institute, so that we can actually discriminate. Are we dealing with West Nile itself, or we are dealing with Usutu, or we are dealing with Zika, or we are dealing with a multiple of them? And that's what we are going to, hopefully, we are expecting those primers anytime now, so that we can work on this. Okay, I just had to show this. This is the immunohistochemistry of the medulla in one of our bats, showing positivity with the West Nile virus in the immunostaining. So all the bats, the pigeons, the cattle egrets that were positive, all of them, we are going to go back and run the PCR, do an RNA extraction in the brains, and go and then find out the ones that are positive, and then we are going to do immuno, and then we are going to find out which area of the brain is actually a predilection site. Do you have it more in the medulla? Do you have it more in the cerebellum? Do you have it more in the olfactory bulb? Or it's a pantropic virus across the brain, because we're a bit more biased for the nervous system in the lab. Okay, so the next phase for the West Nile research is seroprevalence in zoo. So we just got this ethical approval. Well, we've made the correction. We are now going to bleed the zoo workers. Okay, the zoo workers, current workers, and those who have retired in the last 5 years. Okay, so we are going to take their blood sample, and then we're going to check their seroprevalence for West Nile. And all the veterinary doctors that have been posted to the zoo in the last 5 years. So hopefully that should be done. The ethical approval should be out this week, because we've finished all the details. So we'll get that done so that we can relate it together. We've also taken the vectors in the zoo. So the mosquitoes, the flies, so that we can do a complete human, animal, and vector analysis to see the epidemiology of the West Nile. So analysis of the mosquito in the zoo area, and then discriminate among the West Nile family viruses. So I think lastly, look at bird flu in cattle. So it was a big thing last year, March, almost a year. It was March last year when cows in Kansas and New Mexico, dairy cows suddenly stopped milking. They became inappetent. They were not eating. There was no mortality. After about 5 days, 6 days, they stood up. But the veterinarians really didn't have a diagnosis. They took the blood sample. The hematology looked okay. There was evidence of viral disease, but the animals were okay. But within 3 weeks or a month, they had another wave come. And this time around, I think the breakthrough was the dairy farms in New Mexico, out of community service, give free milk to cat owners in the environment, in the village. And 60% of the cats that they gave the milk to died. So now they had mortality. So while they took the blood sample, they sacrificed the cow. They were able to take dead cats to Iowa State. That's the US Center for Veterinary Diagnosis. And when they were posting the cats, immediately the pathologist saw the lung, said, this is an influenza lung. Okay. They took the lung sample, ran the PCR, and so it was positive for highly pathogenic event. So they went back to the cattle sample and ran the lymph nodes, and so it was positive for highly pathogenic. Then they went back to the community, took the milk samples, and the milk samples were very high. So, and that's how they established that. Since then, there has been about 3 or 4 fresh outbreaks of HPA1. About 5 people, humans, have contacted the disease. So it's a big problem that that they are facing now, and that's a wake-up call, because when they said the high titer was in the milk, I said, well, it's influenza. The lungs and the milk, the lungs and the mammary gland are both alveolar tissue. So obviously there is an affinity for the alveolar tissue, and that's why we're not surprised that the mammary gland is being affected. So we did a preliminary screen in Nigeria, because Nigeria imports everything from everywhere, so without regulation. So I was not going to join the train to say we do not have highly pathogenic avian influenza in cattle. We should investigate. And that's one of the mandates of the hub that the Germans set up. So we had these lovely vet doctors who were just finishing their school and they had a holiday before their next career with a master's student. So they went around the country bleeding cattle from the northeast to the central to the northwest. This is where we are. And then we had about 700 samples, 600 and something, and 54 milking cows. We got the milk samples. And what do we have? 31 animals were positive for influenza A, not the highly pathogenic avian, just the presence of influenza. Once the influenza virus is there, you get an antibody response to influenza A. So we got 31, and then we ran the PCR for all the positive, and we took some negatives, but the PCR for influenza A was negative. So that means none of those animals at that time had a current infection, but 31 animals showed evidence that they had contacted influenza before. And then we ran all this sample through an H5N1 ELISA, and it was all negative. So the good news is that there is not any evidence of highly pathogenic avian influenza in the cattle and milk that we looked at so far. No PCR positive, no antigen, but 31 cattle showed evidence that they had responded to one influenza virus before. And so that calls for continuous surveillance. And that even though it looks like good news for now, we are not out of the woods yet. We have to keep investigating. Okay, so 31 of our blood samples were positive for influenza A virus, negative all the milk samples. 54 milk samples were looked at. A study revealed that the detected influenza antibodies were not in response to highly pathogenic avian influenza. Then all the influenza, the gene was not detected in all of our samples. And this is the first report of influenza A virus prevalence among cattle in sub-Saharan Africa. So, like we said, active surveillance hardly goes on, and that's something we are committed to doing. We submitted this also just recently, just last week, to Emerging Infectious Diseases. Influenza A virus in Nigerian cattle. First serological evidence shows absence of H5 subtype and the implications for zoonotic surveillance in sub-Saharan Africa. Okay, so looking into the future, one of the reasons why we came here to give such a lecture is we are looking forward to more North-South collaboration. Things like West Nile, you want to look at migratory birds. They are migratory birds, so they are not only in Europe. So it's good we look at such birds in Europe, in Africa, in North Africa, Sub-Saharan Africa, during the migratory seasons, to look at unusual hosts, squirrels, white rats, birds, pigeons, and go across continents so that we can all be candidates to write big grants, collaborating between Europe, North Africa, Sub-Saharan Africa. We need to update our surveillance data to be things dynamic. When we looked at the PCR for our monkeys, this year was 50% positive. The next year, 1 year after, was 90% positive. So surveillance has to be continuous and to actually get a fact of what is happening in the system. Okay, so we need to update our prevalence status. We need to find out whether mutations are going on and this will help for policy makers to update how we do policy. And eventually, by the time we keep doing our genetic sequencing, we can best help vaccine development. I want to thank you again for listening. I hope I got the spellings correctly. Thank you very much.